Okay, just before I welcome you, I want to add some extra information on the slides of ICO, because they thought that I was going to do everything, but I realized very soon that we needed much more people, much more hands to help us. ICO talked about the insects in Sweden, but they also wanted to know everything about the insects in Madagascar. And we needed someone that had experience in Madagascar. So we invited Brian Fisher, that works at the Madagascar Biodiversity Center. And then he could help us. He has a lot of experience in Madagascar, and he could help us do the work there. But then we also needed someone to look at, and we decided to add another objective. That is not only to look at insects, but why not look at the friends of the insects, so the microbes that live within the insects. And we have Piotr, that is an expert on that, and you will hear some more about that and how they shape insect communities. So we had Piotr joining the team. But then, of course, we needed some very clever people to make sense of all the data. And then we have our postdocs and researchers, Ella, Rob, and Laura, and you will all hear about what they have to say today. And finally, there is many other people that I will just briefly mention. Scarlett, that many of you here that worked as a volunteer know, she was the one who engaged all of you in the project. Dave Carlsson, that he's here, and he allowed us to basically take over Station Linné to run the project, and it was always very kind and welcoming, so thank you so much. David, Daniela, Oscar, and all the other field workers that help us during the peak season to do the ecological measurements. Many people in the lab, both here at the museum, but also at Station Linné and at the Museum of Piotr in Krakow. And I'm sure there are many people that I forgot. But most importantly, today I think it's to thank all of you, the volunteers. I can see many of you here, and with this I would like to welcome you all. Thank you so much for your dedication to the project. Without you, we wouldn't be able to achieve what we are going to show you today. So thank you so much and welcome. So now I'm going to start my presentation. I'll do just a brief overview of the project, and I will pass on to my colleagues to present you the most interesting parts of the results that we have obtained so far. So the Insect Biome Atlas, as Aiko mentioned, was interested in studying and characterizing the insect faunas of Sweden and Madagascar. And why these two countries? Well, Sweden has one of the smallest insect faunas in the world, but it's also one of the most well-known. And when you are developing new methods for serving and identifying species, it's really important to know well what you have, so that you can benchmark your methods against something that you know. And then once you have that, you can use those methods with confidence to explore other underexplored faunas, like Madagascar. Madagascar has a much higher diversity, perhaps a order of magnitude higher, but we know very little about it. So now with the methods optimized, we can explore what we other regions like Madagascar, that is an incredible biodiversity hotspot. But also, at the same time, we have quite a lot of differences in the evolutionary histories of these two biomes. So in Sweden, most of the insects that exist here have come from elsewhere. They have colonized Sweden comparatively recently, after the last ice age. Whilst in Madagascar, all of the species that exist there, they have evolved in situ, they are endemic to the island, and they have been evolving and co-evolving over more than 80 million years ago, since Madagascar got separated from mainland Africa. So this in itself will allow us to make quite interesting comparisons between these two very different systems. So the main questions is what is the size and composition of the insect fauna in these two countries? Frederick will talk about that later. what are the key determinants of insect biodiversity, and you will hear Rob talking about it. But then also we want to know what insects are doing. Insects are key players in the ecosystems, so we want to relate insect biodiversity to ecosystem functioning, and Laura will talk about this as well. And finally, we want to look, just to make it a little bit more challenging, we want to look at the microbes that live within the insects and how do they shape the insect communities. And here you will hear Piotr about it later. So when you want to study insects, this is a very good method. This is a Malaise trap that was invented by René Malaise. He was an entomologist here at this institution. And the first question we had is, where do we set up the traps? So we are lucky enough in Sweden to have long-term monitoring studies that tackle other aspects of biodiversity. And this is the National Inventory of Landscapes in Sweden. They have, over 17 years, they have collected quite detailed information across these 600 plots in Sweden. And they have collected information of more than 300 variables related to vegetation, tree cover, habitat, and landscape changes in these sites. Within those 1 kilometer squares, they have 12 circular squares where they sample in more detail the vegetation, so we can track vegetation changes through time. And we decided, well, this is great. We are going to set up our Malaise traps associated with these plots, because then we can gather information that will be quite useful in the future. So we have set up single traps that cover only one habitat. But we also decided to, we are interested how communities change within habitats, but also when we move several distances. So we decided to collect as well, to set up as well Malaise traps, more than one Malaise trap in each one of these sites, up to 6 Malaise traps covering up to 2 different habitats. Here in this case you have forests and croplands in the lower square. So we set up Malaise traps across all the habitats in Sweden: forests, croplands, wetlands, urban, alpine, grasslands, and a few in the lake shores. This is how it looks like. So we've, it's quite an extensive sampling design. And we couldn't be everywhere at the same time, so we recruited some volunteers that were quite keen to help us. So they visited these traps for a full year between January and December, every weekend during the summer and spring, and every month during the winter. We had 203 of such Malaise traps, 150 volunteers, and we've collected 4,753 bulk insect communities. In Madagascar, we had a different approach. So most of the species in Madagascar are associated with the rainforests, but unfortunately there has been huge deforestation. More than 80% of the forest habitats have been cut down. So we basically what this means for in a biodiversity hotspot is that we are losing species as we speak. So we thought it would be quite important to try to do an inventory of the species that are still there. So we've only focused on rainforests and in the east of Madagascar and the dry forests in the west. Also, logistically, it's quite complicated to sample in Madagascar. There's not much infrastructure to access these protected areas. So we also set up much less traps than what we did in Sweden. So we ended up with 50 Malaise traps. These are within protected areas, so we engage with local community members and also park rangers. So 50 Malaise traps, 104 park rangers, local community members, and here we have collected also a full year every week. So there's no snow in Madagascar. We don't need to in the winter to reduce the sampling. So we've collected more than 2,500 samples. Now, when you have these samples, what we want to know, now we did all of this work, so how do we want to know what species are in each one of these thousands of bottles that we've collected. Traditionally what we do is that we go through each one of these samples, we sort it out to taxonomic groups of the lower taxonomic rank, to families and to genera, and then we send it out to specialists that hopefully will tell us what exists in these samples. But we know from previous studies that this is a very slow process, slow and costly. So we made an estimate, and it will take us more than 1,000 years to be able to identify all the specimens that we have collected. And this, of course, is not good enough. I think our funders will be very upset if we say, well, we are still working on it in 1,000 years. So we need new methods, and this is where DNA-based methods for species identification come into play. Ella is going to talk about this in quite a detail, but I'll just briefly explain. What we do is that we do a DNA extraction in that bottle that we have collected. So the insects never leave the bottle. They leak the DNA with a little bit of magic. We then sequence a gene that is called a barcode gene that is unique for each species. We then pull 800 samples together, each one with a unique identifier, and we massively sequence it. And then in the end, we are able to assign, with some tricks that Frederick is going to talk about, we are able to assign a species, and we know exactly what species are in each bottle. So what is important here is that the animals remain intact. So previous methods used to do a soup out of this, and it was very important for us to keep the animals as intact as possible, because we can go back again to the samples and study and confirm the results we have. New species can be described, results can be reanalyzed. As the databases become better and better, we can reanalyze the data and get more information out of the data. It's also easier to detect rare and small species, and this basically provides us an opportunity to do real-time monitoring of these insect communities, which was not feasible before. I won't go into detail here, Ella is going to talk about it, but in Sweden we also applied other methods to try to get not only the species that exist in each bottle, but also the abundance of each species, which is quite an important ecological data. In Madagascar, we only metabarcoded 75% of the samples, 25%, so one sample from every month stayed in Madagascar to allow researchers there to work on those samples. So basically this is a unique data set. We have the bulk insect communities, so we can learn about the insects, but at the same time we can also learn about the bacteria that live within the insects. And also because insects interact with their environment, we can also learn, we have pollinators that bring pollen into the samples, we have herbivores that have in their stomach plant material, and we have blood-feeding animals. So we can also explore other parts of biodiversity like plants and vertebrates. So it's really an incredible data set. We also, at each one of the sites, we also collected soil samples, soil and litter samples, to metabarcode the arthropods, the bacteria, and the fungi. These both in Sweden and Madagascar. And we also, because we know temperature and humidity are quite important for insects, we attach these loggers to each ballet trap, and we record every 4 hours temperature and humidity. That will be important for to study the drivers of insect communities. And finally, we measured ecosystem functions, things like herbivory rates, pollination, seed dispersal, decomposition, and so on. And then within Sweden, we have this huge data set that I just talked to you about that we can also tap into. For Madagascar, we don't have this data, so we collected this data ourselves. In all sites, we collected data on standing characteristics, vegetation. So basically, the results, we have more than 7,000 bulk insect samples. We estimated, we counted some samples, and we have estimated that we have 7 million specimens in Sweden and 1.7 in Madagascar. 90% of the material, more than 90% of the material was processed via metabarcoding. We got 700,000 ASVs, these are these unique barcodes for Sweden, and the same for Madagascar, but be aware that Madagascar has 25% of the samples that we have in Sweden, so it's much more diverse. And then when we group these unique barcodes into what we call OTUs, operational taxonomic units, which is the equivalent of species, we get almost 34,000 species in Sweden and 77,000 in Madagascar. From these species, we can add species names in Sweden to about a third of this, and to Madagascar to less than 3%. So this means that thousands of species have never been sequenced before. Many of these are also new to science. Basically, we have collected more data on Swedish insects than 300 years of research since Carl von Linné. And if you remember my last slide, now you can do what was, you will need 1,000 years, we can now do in 1 year. And in fact, you can do it in 1 month if you increase your lab capacity and your lab technicians. So that's it. This project has been funded by the Knut Alice Wallenberg Foundation, but a lot of institutions, both in Sweden, in Madagascar, and Poland and the United States have been involved. So it was really a huge effort and a huge collaboration. Thank you.